Right now in May 2026, there are two separate outbreaks of extremely dangerous viruses happening at the same time. One you've probably already heard about. It's the Hanta virus outbreak on a cruise ship called the MV Hondius that sailed from Argentina. 3 people have already died from that, and then just yesterday, health authorities in Africa confirmed a new Ebola outbreak in the Democratic Republic of Congo in a region called the Ituri province. 246 suspected cases and 65 deaths. And to make it worse, early results suggest that this might be a strain of Ebola for which the existing vaccines don't even work. So we have 2 deadly viruses, 2 active outbreaks happening simultaneously. And that's why I wanted to make this video, because I think it's actually a really good moment to explain what it is that actually makes certain viruses so much more dangerous than others. And there are 4 specific factors that scientists look at, so let's go through them. Both hantavirus and Ebola are what's called biosafety level 4 pathogens, BSL-4. Now that's the highest category that there is. When scientists work with these viruses in a laboratory, they have to wear a full pressurized suit with its own air supply. You've probably seen the pictures and also movies about that. The whole point is that the level of protection is because these viruses are so dangerous that even a tiny mistake can be fatal. And the reason that they sit at this highest level comes down to these 4 factors that I will talk about. The first factor is called infectivity. And this basically comes down to one question. How many virus particles does it actually take to infect a person? With some viruses, your body needs to be hit with a very large number of them before infection can get established. With others, it only takes a tiny amount. Norovirus, for example, it's the stomach bug that tears through cruise ships, only takes somewhere between 10 and 18 individual virus particles to infect you. Now, that's almost nothing. Ebola is in the same category. Studies have shown that fewer than about 10 viral particles are enough to cause an infection. And now with hantavirus, so we don't have a precise number because obviously you can also not do any experiment on people. But what the epidemiology tells us is that people develop full infections from spending just a short amount of time in a space where infected rodent droppings have been distributed. Now that tells you the number must be quite low, the number of virus particles, even without a lab figure attached to it. Compare that to HIV, that's the virus that causes AIDS, where the infectious dose is actually quite variable. It can range from a very few particles under certain conditions to tens of thousands even, depending on the source and the route of exposure. Now, and that is one of the reasons why HIV does not spread nearly as easily as people sometimes assume. So let's move on to the second factor. The second factor is called virulence, and this is completely separate from infectivity. Virulence is about how severe the disease actually is once you are infected. A good way to think about it is the common cold. The virulence is quite low, your immune system is able to handle it, you feel rough for a few days, maybe a sore throat, a runny nose, and then it's over. You don't end up in a hospital in most cases. Nobody dies from a common cold. Now look at the other end of the spectrum. The Andes strain of the hantavirus, that's the one that they have found now on the ship, has a case fatality rate of somewhere between 35 and 50%. Ebola is between 25 and 90% depending on the strain and access to care. Now these are not small numbers, and that means if you catch it, there is a very real chance that you will actually die from it. The disease progresses fast, it hits hard, and your body often simply cannot keep up. The third factor is transmissibility, and this is where people often confuse things because it sounds similar to infectivity, but it's actually measuring something completely different. Infectivity, remember, is about how many virus particles you need to be exposed to before an infection gets started. Transmissibility, however, is about how easily those particles get from one person to the other in the first place. Scientists measure transmissibility with something called the R0 value. That's the basic reproduction number. It tells you how many people, on average, one infected person will go on to infect others. If a virus travels through the air, the R0 can be enormous. Measles, for example, has an R0 value of 12 to 18. This means that one person can infect up to 18 other people. That's extraordinarily high. Ebola, despite being so deadly, actually has a relatively low R0 of around 1.5 to 2.5 because it only spreads through direct contact with body fluids. You need blood, vomit or saliva to get into your system, and then you also get the virus. And this limits how fast it can move. HIV that's similar. It requires very specific routes of transmission. And then there is also the hantavirus, which we just talked about, which normally you can only catch from rodents. And now what makes the Andes strain, now that's the one on the ship, what makes this one particularly concerning is that it's the only known hantavirus that can also spread between people through close prolonged contact. And in 2019, an outbreak in Argentina, before any containment was in place, its R0 was measured at just over 2. It's not an enormously high number, but for hantavirus, that's alarming. Once isolation measures were brought in, then the number dropped below 1 and the outbreak then ended. The fourth and final factor is whether there are any countermeasures available. Vaccines, antivirals, treatments, and so on. And this is where both hantavirus and Ebola are in a very difficult territory. For hantavirus, there is no specific antiviral drug. There's no vaccine. Doctors put you into an intensive care unit in ICU to support your breathing and hope that your immune system can do enough. For Ebola, there are 2 vaccines, but both of them target only the Zaire strain of the virus. The outbreak currently unfolding in Congo appears to be of a different strain entirely, which means those vaccines probably don't help. Now compare that to something like measles. There's also no cure, but we have a vaccine that works so well that in vaccinated populations, the disease is essentially eliminated. Or HIV, there's also no vaccine, but we have antiviral drugs that allow people to live quite normal lives. And people have basically an effective treatment options, and their viral load is actually so low that they cannot even transmit the virus easily to others. So the presence or absence of countermeasures completely changes how dangerous a virus is in practice, even if the underlying biology has not changed at all. So when you put all of these 4 factors together, a very low infectious dose, extremely high virulence, transmissibility, which is not so low, and also no specific treatments available, well, that is why both the hantavirus and Ebola, why they sit at the very top end of the danger scale. And that's why they're biosafety level 4. And that's also why health authorities around the world are now watching both of these outbreaks very carefully right now. So that's all I want to share with you today. I hope you found the video interesting. There is a comment section below. I would like to invite you, of course, to leave comments behind. From my side, that's all. Happy microbe hunting as always, and hope to see you in the next video. Bye bye.